You're looking at a slide. This is a slide of one of my favorite books, Body Electric. It's been a very inspirational book, and I'm going to credit the inspiration for this video to my friend Daniel, Mr. Daniel Lynch, who has a great company. He's helping patients on a daily basis to obtain reimbursement from healthcare companies when they do natural medicine. So the subject is the inspiration for my regenerative medicine approach. I told Daniel that electrons and negative energy were very important for salamanders to regenerate, and I'm going to share with you how I know that information. And what's really neat embedded in this talk will be a lot of the fundamental basis of my regenerative therapy, and what I like to think really sets me apart from anyone else who does regenerative medicine or stem cell medicine. And this is going to set apart my future, because you'll see what kind of giants I'm standing on the shoulders of in terms of their research and body of knowledge. So I'm going to go one by one through different pages I've excerpted from this phenomenal book, Body Electric. And I have been able to obtain the ability to send this out for anyone at $5 a copy on a PDF. So you can contact me directly if you would like a copy of this. Now let's go to the next slide. So this is about page 21. I'm going to read directly from Dr. Becker's words. I'm going to use my mouse and pointer. Dr. Becker was an orthopedic surgeon in Bellevue and New York City. He said in his writing, "It's no wonder that medical biology is afflicted with tunnel vision. We know a great deal about certain processes, such as the genetic code, the function of the nervous system, in vision, muscle movement, blood clotting, and respiration. These complex but superficial processes, however, are only the tools life uses for its survival. Most biochemists and doctors aren't much closer to the truth about life than we were 3 decades ago. As Albert Szent-Györgyi, the discoverer of vitamin C, has written, we know life only by its symptoms. We understand virtually nothing about such basic life functions as pain, sleep, and the control of cell differentiation, growth, and healing." I have the rest of this here if anyone wants to read more, but I'm going to skip around. My research began with experiments on regeneration, the ability of some animals, notably the salamander, to grow perfect replacements for parts of the body that have been destroyed. All right, so that's the start. So now this page is outlining this gentleman, Dr. Spallanzani. He discovered the reversal of plant transpiration between light and darkness. In 20 years of meticulous observation, he studied regeneration in worms, slugs, snails, and salamanders. He set new standards for thoroughness, often dissecting the amputated parts. His most important contribution to science was his discovery of the regenerative abilities of the salamander. A young salamander performed the feat of regeneration 6 separate times in 3 months. So that's what that gentleman discovered. There's page 38. Here's page 46. Beautiful slide. Shows the essence of stem cell differentiation. There's a stem cell. I'm going to use my mouse because it'll be important. All gene types available. Then gene expression causes it to develop into all these different aspects. There are details here I'm not going to get into. If you read the book itself, you'll be able to get into these details, talking about mechanistic outlooks, dedifferentiation, one-way, two-way paths. There we go, one-way street. You'll have to read this directly, but it's a wonderful graphic. In this excerpt from page 48, I outline some really important points Dr. Becker's making. Still about the salamander and regeneration, and clearly my theme here is, how do I use this knowledge in my patients and my practice? So now we're here on the salamander directly descended from the evolutionary prototype of all land vertebrates. It is a marvelously complex animal. Its forelimb is basically the same as ours. All of its interrelated parts grow back in proper order. The same day the limb is cut off, debris from dead cells is carried away in the bloodstream. Then I skip down here. This process, exquisitely beautiful and seemingly simple, is full of problems for biology. What organizes the growth? What is the control factor? How does the blastema know that it is making a foreleg instead of he meant hind leg. Now this is cool. The salamander never makes a mistake, unlike the editor of this page. Once again, I outlined some areas I wanted to point out. I love this thing. In order to answer this concept of how does a salamander do this, Dr. Becker goes into an area that I'll make another video about called the morphogenetic field. It's also called morphic resonance by a very intelligent biologist who has a PhD named Rupert Sheldrake. And morphogenesis means the origin of a form, and the field idea is an attempt to get closer to the control factor by reformulating the original problem. A Danish biologist working on regeneration and common flatworms discovered more information, and it's outlined here in this paragraph. And then I'm gonna read the list. The idea grew out of early experiments by Weiss. See, if regrowth clearly wasn't a simple matter of a truncated muscle or bone growing outward to resume its original shape. Structures that were missing entirely, the hand, the wrist, the bones of the salamander, reappeared. So, morphogenetic field. Somehow, there's a field of energy and it helps dictate limb growth after amputation. So here at page 55, the things I outlined with arrows, you can see the salamander in the cut and how the differentiation occurs. So what did they find was important? Nerve connections. In 1823, Tweedy John Todd found that if nerves into the salamander's leg were cut, the limb wouldn't regrow. He got normal regeneration when he gave the nerves time to reconnect before severing the leg. Science was not ready to make anything of this observation, but many experiments since have confirmed this. So this next page is interesting because in the 1940s, they were trying to figure out ways to not let the frog legs regenerate. He tried dipping the wounds in saturated salt solution to prevent the dermis from regrowing over the stump. It worked. Even though the replacements are incomplete, there was a tremendously important breakthrough. The first time any regeneration had been artificially induced in an animal normally lacking such ability. And then Dr. Hay at Cornell in New York City at the medical college, ironically where I trained, she studied the neuroepidermal junction with an electron microscope. You'll have to read page 57 to see what she found. Good, here we are on to page 61. Some really neat observations. A Russian doctor in a 1958 paper from Leningrad, he discovered that there was standard injury in each muscle, and he measured the current of injury, noting the effect on current after injury, and noticed that current decreased with each succeeding nerve cut. The current of injury was proportional to the amount of nerve left behind. So Dr. Becker then had enough pieces to start on the puzzle. He summarized the following, which is getting to the crux of this, that the extent of injury is proportional to regeneration. The amount of nerve is proportional to regeneration. Therefore, the current of injury is proportional to the amount of regeneration needed. So I only have 4 more little pieces here. This is fascinating because this Dr. Rose would prick limb stumps of, I guess he was working with mammals. He saw that making the injury worse could make regeneration better. That's very interesting because I explained to a lot of patients, ozone works by sometimes making injuries worse at first. That's how it works. Singer first confirmed that cutting nerves in a salamander's legs at various stages of regrowth Recent research had found that a salamander could replace its leg if all the motor nerves were cut, but not without sensory nerves. So for my friend Daniel and anyone else reading, here we're getting to the important conclusions. The Russians are amazing researchers. This I got to point out, they're willing to follow their hunches, unlike what we do. Their researchers would get government money to try the most outlandish experiments, ones that our science just knew couldn't work. So he would cut branches from different tomato plants, and here we go, Daniel. A stream of electrons was found flowing from the wound. A similar current of injuries emitted from all wounds in animals. I outlined this here. Then basically battery assisted plants. So now anyone can start understanding. You can look an ampere, electric current. You can start understanding why in my practice I value sound healing, that type of energy, electrons through ozone, and PMF magnet therapy. Couple more slides. We're finally at the kicker. At one point, Dr. Becker finally did his experiments, and here I put a red box around. Salamander can regenerate, the frog can't. So after amputation of each animal, their voltage goes very positive, which is bad because we're saying negative voltage is what we want for healing. But after that period of time, look at the salamander who does generate. Negative voltage, as if he's having internal PMF, internal stem cell therapy, internal electron donation, internal ozone. Boom, that's his regenerative capacity, and the frog cannot regenerate, and that's similar to how a human would be. So we need to mimic this current, this curve of healing through electricity. So this is my final graphic in conclusion. So here you can see there must be a continuous current. Here's a little tiny cute salamander healing. There's the electrons being donated, measuring it. The negative potential seemed to bring forth the all-important blastema, which is the start of healing. The current of injury is more than a side effect. They finally wrote up these publications, and this is a great book. You'll have to read it sometime. But there is the concept of electron donation, electricity, and how it relates to salamanders and what I do in my regenerative medical practice. So I'm very convinced the ideas that I have are going to continue to improve, and I'm going to be looking forward to seeing patients heal from very severe injuries such as spinal cord injuries, autism, maybe seizure disorders, serious musculoskeletal injuries, and cancer, and viral insults. And I'm very happy that these type of publications lay the predication to what I do now. Thank you very much for watching and listening. Have a great day.